Hi everyone, thanks for joining me. Today we are going to talk transfection. Not only how to perform transfection and what it is, but what do you do when your transfections aren't working? And our transfection talk today is brought to you by Mirus Bio, home of the transfection experts. Alright, so let's get started. For all of you non-transfection experts out there, let's first define what transfection is. Transfection can be broadly defined as the delivery of exogenous molecules into cells. And another way of putting that is alien or foreign molecules being delivered into cells. And why would you want to do that? Why do we want to introduce DNA, RNA, or protein into cells if it wasn't already there? There are many reasons why we would want to do it, but let me just list a couple of them in case this is what you do in your lab or what you would like to do. You might want to transfect Cas9 to use CRISPR to modify genomes, or you might want to use your cells as factories to produce virus or protein for therapeutics, and you might want to overexpress genes or knock them down in order to understand cellular mechanisms. Your reasons might differ, but delivery of foreign nucleic acid, transfection, is a very powerful tool for both basic and applied research. And there are many methods of transfection, but the one you might most frequently run into is using chemical transfection reagents, like our Transient reagents for transfection. So I'm going to stop talking for a bit here, and let's sit back and watch this short video clip on how transfection happens with transfection reagents.
How do we transit? The process of transfection begins with transfection reagents that coat and condense the negatively charged nucleic acid into a transfection complex with a net positive charge. Transfection complexes are then incubated with target cells to allow binding with the negatively charged cell membrane, followed by uptake through endocytosis into cellular vesicles called endosomes. The transfection complex must then escape the endosome's acidic environment and be released into the cytoplasm. Nucleic acids such as siRNA, microRNA, and mRNA can function right here, whereas plasmid DNA must be shuttled into the nucleus for transcription into mRNA to eventually produce target protein. At Mirus, we care as much about transfection as you do about your research. As a result, Mirus develops transit solutions to provide better performance and lower toxicity than other reagents.
Alright, I hope you enjoyed that video. The ultimate goal of your experiment might differ, but what we generally want in our transfections is for them to be efficient without killing our cells. And as many of you might already know, striking that balance is not always easy. Yes, if cells just took up random material from the environment all willy-nilly, we would all be mutants. Then, a question that we often get is, how long does transfection or gene expression post-transfection, how long does that last? Is it forever? And it depends. You might have heard of these terms before, transient versus stable. And it's exactly what it sounds like. Essentially, transient transfection isn't permanent. The transfected material, it doesn't integrate into the host cell's genome, so it doesn't replicate when the host cells divide. And as a result, that expression is short-term. For general protein expression off a plasmid, that could peak around 48 to 72 hours post-transfection, versus in stable transfection, some longer-lasting change or integration into the host genome happens, and transfection results in a stable expression. Most of the time, whether you're using chemical or electroporation-based transfection methods, expression is going to be transient with a very, very small percentage of your transfected cells integrating the plasmid into their genome. And there are protocols for selecting these rare events, but often other methods are employed, such as virus-mediated transduction or endonuclease-mediated genome editing to permanently edit a cell's genome and create a stable cell line. So let's look at this example here where virus is being produced through transfection. In the first step, usually in HEC293 cells, virus is produced by transfecting plasmids that encode for the viral genes and viral proteins. And virus is then collected. So in step 2, you see that happening. And that collected virus can then be used to infect cells in a process known as transduction, what's shown in step 3 there. And depending on the virus, once the cell is infected, the virus can integrate its packaged genome into the host genome. And the cell can then express stably that viral gene, which is often your gene of interest. So if you're having trouble directly transfecting your cells and selecting for stable expressors, you might want to consider an approach like this, using lentivirus-mediated transduction to create a stable cell line. And it's the same with CRISPR-Cas9 and other genome-modifying enzymes. Transfection is one of the first steps. You need to introduce those modifiers somehow, so you can do that by introducing plasmid or mRNA that encodes a protein, or the protein itself in some cases, like with Cas9. Often Cas9 ribonuclear protein complexes are transfected. And okay, that's all fine and well, but the title of this talk is How Do I Transfect My Dang Cells, and what to do when it doesn't work. So in the next part of this talk, let's go over how it's done in the lab, practically speaking. At least with the Transient reagents, the protocol is super simple. You first want to mix your DNA or whatever nucleic acid that you want to deliver into serum-free media. Then you're going to mix in your transfection reagent. And step 2 is pretty easy. You just sit back and wait. And it's important to wait and let the complex incubate, as it takes time to form. And the amount of time that you're going to wait, that's going to vary by the reagent you're using and what you're using it for. And the next step, you're going to add that mixture gently to your cells. So what I mean is, if your cells are on a plate, don't blast off the cells with your pipetter when you're adding the transfection complex. Then you want to evenly distribute the mixture by, dropping it evenly over the plate, but also gently shaking or rocking the plate side to side, backward and forward. And the last step, the best step, you want to check and harvest your cells as necessary. And like in this example, hopefully you come into lab and what you see is nearly 100% of your cells are expressing the protein that you want. In this example here, these are stem cells, iPSCs, that were transfected with the GFP encoding plasmid. But raise your hand if instead this has happened when you come into lab the next day. You look at your cells and they show no signs of transfection, or your cells are dead, or even worse, they are dead and show no signs of transfection. It's very sad. Or maybe you have some less dramatic version of the above. Okay, so in the next part of the talk, let's cover some transfection parameters and troubleshooting tips for how to get the best transfection efficiency with the least cytotoxicity possible. Okay, one of the most important factors to a successful transfection is the state or the health of your cells. Especially for gene expression off a plasmid, you want your cells to be happily dividing, as it's thought that during cell division, the plasmid is taken into the nucleus when the nuclear envelope breaks down. So let's look at this first question. Does cell density or confluency matter for transfection? Heck yeah, it does. These are 3T3 cells, by the way. Whatever the cell type, though, the cell density at the time of transfection can have a big impact on how your transfection results. In this example, a luciferase plasmid was transfected into 3T3 cells that were either 75% confluent, that's the red bars, or they were 100% confluent, covering the plate entirely, that's the gray bars. And as you can see there, if the plates were covered and the cells were not dividing, the luciferase expression was much lower. And so this is just for this particular transfection. The optimal number of cells or confluency or density you want is going to depend on the cell type that you're transfecting and what you're transfecting for. Okay, and you might have noticed here there are 3 sets of bars per series, and that's because we also looked at the volume of transfection reagent per microgram of DNA that was transfected. And as you can see, for this particular experiment, 3 microliters was optimal, 3 microliters per microgram of DNA. And the volume of reagent used per milliliter of culture, it can have an impact on not only the efficiency, but also the cytotoxicity that results from transfection. If you can get the same amount of transfection with less reagent, that's always better for reducing cytotoxicity. You can also change the media 4 to 6 hours after transfection. We think that it's thought that most of the complex is taken up at that point. We don't usually recommend it though, because most cells, they don't benefit from a media change. Some cells, they actually do like having some fresh media, but it's all dependent on your experimental system. All right, so we talked about the state of the cells. How about the state of the thing that you're delivering? Does nucleic acid quality matter? Yes, it does. So take a look at this graph here where we're showing you luciferase expression when we transfect into COS7 cells a plasmid that was either commercially prepared or one that was what we call a crude plasmid from a mini prep kit. And oftentimes these kits, they don't do a good job of removing contaminants like endotoxin. As you can see here, the expression of luciferase was much lower in that crude prep versus the clean commercial prep. The third bar there is that same crude prep after it's been cleaned with our MiraClean treatment. And what's important about this slide is you want to be sure that what you're transfecting is what you think it is, that it's good quality. So you can do something like sequencing or running your plasmid on a gel to ensure that. Next, does complex formation time matter? Does how long you incubate your nucleic acid with reagent, does that matter? And yes, it does. Over time, as you can see in this graph here, the size of the complex grows. So that's what's plotted on the y-axis, the complex radius in nanometers as measured by DLS. Over time, you can see that the complex is growing. And you can imagine that if the complex is too large, it's not readily uptaken by cells. So you want to follow what your protocol suggests for the incubation time of reagent and nucleic acid. Okay, and you also might have noticed on this graph that there are red dots and black dots that correspond to transfection complexes that were formed at various microliters to microgram reagent to DNA ratio. Kind of discussed this already, but notice that the size of the complex, the growth, also depends on that ratio. So again, that ratio does matter for transfection efficiency. The optimal ratio, it varies by both the cell type, reagent, and also application. So keep that in mind. How about transfection reagent formulation? Does it matter which reagent that you use? And you know the drill. Yes, it does matter. Shown here are adeno-associated and lentivirus titers from transient transfection using Transit virus gen, the red bars, or other commercially available reagents. And as you can see, highest titers were obtained using Transit virus gen. And that might seem like a gratuitous plug for the Mirus brand, but even within the Mirus catalog, there are reagents that perform better than other Mirus reagents, and it really depends on the circumstance. Within the Mirus catalog, there are broad spectrum reagents like Transit X2. Transit X2 can deliver plasmid, siRNA, even Cas9 RNPs. There are reagents for electroporation, as well as reagents for specific applications like virus production with Transit virus gen and Lenti. We also offer turnkey solutions for cell type specific transfections, such as those listed there, and reagents for mRNA or larger RNA delivery. and reagents for oligos like siRNA, and also reagents for protein production in high-density suspension CHO and 293 cells. All right, so we're getting to the end of our time here together, and I just want you to remember, cells have feelings too. Just look at that guy. So each experimental system is unique, and when your transfection isn't working, or if you're just starting out, you might want to adjust some of the parameters that we discussed today. You can visit our website. The link there brings you to this optimization protocol, which outlines testing different reagent to DNA ratios that we talked about. When transfecting a new cell type or nucleic acid, we always recommend that you test a positive control nucleic acid and also a matrix of conditions on the small scale if possible. And once you identify the best conditions for your given cell type, application, nucleic acid, then move on to your more precious nucleic acid and experiments. Also available is our reagent agent transfection database, which you can find at mirasbio.com/RA. Reagent agent is really easy to use. All you have to do is input a given cell type and nucleic acid that you're interested in transfecting, and it will spit out, when available, conditions that we have identified that are optimal for that combination. In addition to reagent agent, you can request samples on our website at mirasbio.com/sample. We are always happy to provide multiple reagents for you to test, so you can pick out which one might work the best for a given cell type or application that you are working on. So unfortunately, I did not have time today to discuss all the facets of transfection troubleshooting. However, let's quickly run through a list of questions that you can ask yourself when you're troubleshooting. What cell types are you using? So some cell types are just inherently difficult to transfect via chemical transfection. So in those cases, you might want to consider an alternative method like electroporation or transduction with virus. Have you tried transfecting a different or fresh batch of cells? Sometimes cells that have reached senescence at a high passage number, they aren't the best at being transfected. So you might want to thaw out another aliquot of cells. Volume of reagent and amount of DNA, confluence of cells, we already discussed those. What method do you use to assess transfection efficiency? Okay, so what I mean by this is that your transfection, that actually might be going pretty well, but the way that you're measuring whether it's going well or not, maybe there's an issue there. How long after transfection do you assay the cells? So this is particularly important for something like knockdown, where at the RNA level, the RNA might be degraded, but the protein that was already existing pre-transfection, that might not have degraded yet. So you might want to assess your knockdown maybe 48 to 72 hours post-transfection rather than 24. What medium do you use for cell growth and complex formation? So we kind of touched on this, but you don't want to have strange additives in the medium so that it might interfere with the formation of those complexes. Have you performed a cells alone or a reagent only transfection? So these controls are particularly important to identify where cytotoxicity is coming from. Size of the plasmid, yeah, so large plasmids are indeed harder to transfect, and you can try increasing the dosage of the transfection complex that's being delivered in order to deliver more copies of the plasmid. So what I mean is, if you found that a 3-to-1 ratio works well, try upping that to a 6-to-2. So that's still a 3-to-1 ratio, but you're just doubling the amount of complex. And purifying DNA, using the construct successfully in another cell line. So really with these, what you want to do is confirm that your plasmid prep is good to go. mycoplasma contamination, and other changes that have been made since your last successful transfection. If your transfection, you've been doing the same one over and over and everything's been fine and suddenly it just stops working out of the blue, these are questions that you might want to ask. Some simple changes to your transfection, including change of brand in tissue culture flask or different lot of FBS, they can change the way your cells grow and thus affect transfection efficiency. And lastly, have you compared the performance with another reagent? So check with a neighboring lab or dig around in your refrigerator and see if you have a different reagent that you can compare to your current reagent that you think is the issue. And this might help eliminate the reagent as the root cause of your problems. All right, so if you've exhausted all those options and still nothing is working for you, or if you just want to chat about transfection, always feel free to turn to Mirus Bio. So you can do that by shooting us an email at the address there or giving us a call. We are always happy to help, like I said. So if you're watching this live, we are happy to take any of your questions right now. Thanks.